Okay, so we've already probably set a record as far as the number of times you've heard the words targeted, untargeted, and fluxomics within an 8-hour frame of time. But guess what? You get more. So the way we structured the talks this time was as Barb was telling you, we have the workflow that you would experience with the metabolomics experiment. So we started with sample preparation, relative things, and then relevant things, and then interacting with the core. And now we're going to tell you the details of the biological experiment. The only problem with that is, in order for those people to tell you how you interact with the core or what you're going to do as far as your sample preparation, they had to tell you about a lot of this stuff already. So hopefully this isn't excessively boring. My goal for you with this talk is that you'll be ready by the time you're done with this and having heard it for the third time, and maybe in a little bit more granular detail about the specifics of the steps rather than the steps in the context of an example or anything else, that you'll be ready to say, okay, I'm going to go do my experiment, and I'm going to consult with the core, of course, but what I expect that I want to do is an untargeted experiment, and I know why. And if you don't get to that point through this talk, then stop me, say, hey, this just isn't making sense, or I need clarification on a certain point, because that's our goal. So along those lines, at the end of the session, understand what exactly a metabolite is. Okay, well, we've been talking about metabolites all this time. No one's really defined them yet. Most of you have a good sense of what this is, but I like to, in the context of this talk where I'm talking in granular detail about classes of metabolomics, I like to define it in my terms, and then when and how metabolomics is useful, so that you can know when to do it and when not to do it. We're gonna go in detail and break down these three techniques in a sort of a classroom manner. We'll describe the purpose and limitations of each technique, and then we'll give some examples. You've already seen good examples of all of them, and hopefully this talk will cement that in your mind and give you some additional perspectives as well. So defining terms, we'll overview metabolomic strategies, targeted, untargeted fluxomics. Already basically said all of that. Okay, where are we in our workflow? We are mostly here. In the experimental design sort of things, but it affects all of these steps down the road. Okay, what is a metabolite? Okay. If you don't know already, the easiest way to define it is to define what a metabolite is. What a metabolite is not. It is not DNA. It is not RNA. It is not protein, but it is produced by a biological organism. A small molecule, typically molecular weight of what we're talking about is less than 2,000. That's some peptides would be under that category. This will include many different categories: fuel, substrate, a structural component of a cell, a signaling molecule. Many other classes, exogenous compounds, can be metabolites, can become involved in the endogenous metabolome, but there's a lot of interplay there. And then, of course, the things that are not metabolites interact with the metabolites, or are made up of metabolites. Deoxynucleosides are a component in DNA, nucleosides in RNA. Peptides are part of proteins, but those are also amino acids ultimately make those up. So there's exchange between all of these categories, and so you see that the omics, metabolomics is not a separate and unique island as compared to the other omics sciences. It's integratively connected through many pathways. Okay, and then what is metabolomics? Well, you probably wouldn't be here if you didn't already know this, but just to set a nice definition to it, or a definition to it, in case you're wondering as we talk about it, the attempt to identify and quantify all of these compounds, or some fraction thereof, in biological samples enrolled in biological systems. Okay, so this is useful not because you haven't seen this workflow diagram, but because it helps define in context who's going to do what in these types of experiments, for most of you. Some of you may be involved in other parts of this. So you may be getting confused when we start talking about sample preparation. Okay, you're talking about extraction solvents. Maybe you're talking about analytical methods a little bit tomorrow. What of that do I have to worry about? What of that is going to be handled by the core lab? And this is how I sort of break it down. The biological experiment is going to be handled by you in collaboration with us, the core lab. We want to get involved as early as you feel is necessary, and as early as is feasible, to make sure this happens right. Sample prep, you may do some of the aspects of sample prep. We talked about sampling of tissues. That's part of the experiments that are typically done by harvesting of cells, collecting of tissues. That's all part of sample prep. Extraction, that's generally done by us, so there's a transition over. Exactly where that happens will depend a little bit on the assay that's being performed. Instrumental analysis, the proverbial black box, is what mainly is done by us. We'll get into that black box tomorrow. And then it'll transition back rapidly to data analysis interpretation, which is done in part by you, but with the Metabolomics core here to help you and assist you and give you the tools that you need to see that. And so let's see. I'll hit the button again. I'm sorry. I didn't mean to bring it up. Yeah. Now it's black. It wasn't black on. OK, here we go. It's not black on my screen here. I'll do it again. I'm pointing. Don't touch it. No, I know it. That was, well, all right. Single click. Waiting. I am sorry. I'm
sure it's not your fault.
No, it's probably my fault. There we go. Needed somebody else's hands. I'll set this down for now. Okay, very good. So that's the workflow. Okay, targeted and untargeted. Again, I apologize for the repetition. If it is repetition, for those of you who like this sort of clarification, hopefully this is helpful. So targeted is what you do if you know what you're looking for, right? You've already got a sense of what pathway you're interested in, and you want to go after it. Generally, a targeted experiment is limited to tens or hundreds of compounds at a time. As Maureen said, she's sort of taking a little bit of a expanded viewpoint on this. If we're going to do an extraction for you and run samples, we want to give you as much as is available for your money. So if there's a class of metabolites that sort of comes along for the ride with an extraction, the core is going to do its best to give that to you, even if what you really are focused in on is glycolysis and TCA metabolites. Now that may mean that the sample preparation is devised to be specific to that particular class, and that's what you're really guaranteed to get. And the rest of it is with some slightly higher level of uncertainty. But the goal is that you're going to get better information about that class, and then as much as you can elsewise. But it's usually not going to be thousands of compounds, right? We're targeting a particular class. The good thing about targeted is that you're going to get confident identities. You don't have to worry about, is this am I sure I know what metabolite I'm looking at? Am I looking at some other artifact or some weird feature that's been incorrectly or uncertainly identified? And the reason is because we base our identities on, okay, I didn't touch anything this time. Did I walk too close?
Maybe stepping on something.
I don't know. Something is weird here. Let's check our connections. I don't know. Let's just skip that thing.
You can use it as a pointer.
That's perfect. Okay, great. So metabolomics approaches. So yeah, so everything's confidently identified because it's based on authentic standards, and you can do either absolute or relative quantitation. We can measure these things at nanomole, micromole level if you need it. And an example would be a free fatty acid profile of a blood serum. Untargeted is what you don't know when you're looking for, or you want to look at everything. You want to say, I want as much coverage of this sample as I can get. It can give you up to thousands of compounds depending on the samples that you hand us and the sensitivity of the methods that we use. Okay. I didn't touch anything again.
They sent the projector up.
Yeah, that's what I'm worried about, is that we could be looking at that. The screens down here are blank. Why is it flashing on and off? Some signal that I've got here. I'm sorry. Yeah. You want to switch over to our backup? Let me check these cables here. It could have been this little what you call guy there could have been.
Okay. Just Pretend like it's going to be okay.
All right, I'm going to pretend like it's going to be okay and keep moving because we've got limited time and you guys hopefully want to learn what we're looking at. The advantage is, so we can get lots of compounds. That's good. The disadvantage is that it has less confident identities. Not everything that you have in an untargeted analysis will be confidently identified. We'll give you as many confident identities as we can based on our library of known metabolites that have been run on the method. But there's going to be additional features there that won't have an ID with them. And so we'll talk about what you do with that. Generally, untargeted analysis is limited to relative quantitation. There is, some people have said, oh, this is not quantitative. That's not true. We can get a good sense of what changes, and when you correlate it with a targeted analysis, usually you come out with very, very similar results. But we can't tell you in a nanomolar quantity in general what's going on. All right, and then an example would be a global study of the metabolic response to change in a diet is what I had on there. Thoughts and ideas? We'll just keep going, and I'm going to pretend like it's not happening, and I apologize for while we work on our backup options, unless you tell me to stop. So targeted metabolomics workflow. Here are the steps, and we're going to break down these steps in a little bit of detail so you can get a good sense of what's going on. There's sample preparation. Then there's instrumental analysis. We'll talk more about that in detail tomorrow. Data analysis. Maureen touched on that a little bit. We'll do calibration and quantitation, and then data interpretation. And so these steps are relatively straightforward. So here's sample preparation. The goal of sample preparation is to make a biological sample ready for analysis on an instrument. And if we wait for our stuff, the goal is, so basically the steps that you're going to do are you're going to use an extraction solvent to remove the metabolites from your cells. And you're going to then remove the residual debris from your samples by centrifugation or filtration or something like that. And what you should be left with is a solution containing your metabolites of interest and not containing any of the stuff you don't want. There's various different extraction solvents that you can use. Generally, they're an organic solvent. They might be any number of different things. TCA, I heard someone mention that as a typical extraction solvent for metabolites that used for biochemical assays. It is not as widely used for metabolomics because it's not as compatible with mass spectrometry, and it may contain internal standards. So sample preparation. So this is a sort of an animated slide. After your metabolites are extracted, what's next? Okay, good. So we're getting our other projector up and running. I will let that go before we continue. And
We also have this on our Dropbox.
yeah, that's true. I've got a real small screen here, so if you guys are looking at it, I'm fine with moving forwards while we wait. But it's up to you what we do. Recommendations from organizers. Everyone comfortable with me just plowing ahead and staying as little distracted? Okay. So after metabolites are extracted, what is next? So the goal is to get from your sample to your instrument. And one possibility is you just analyze your extracted sample as is. You inject it on the instrument. And that's really nice when that works out. The other option is that you may need to dry down your samples and reconstitute it in a different solvent to make it compatible with an instrumental analysis. So we'll do that if we need to, as long as your samples are stable to that, and the core lab will hopefully have set up that appropriately. Another option is that you might need to further purify your sample. So if you're looking at a very low level of metabolite and it's of particular importance, what we may want to do is selectively enrich a particular class of metabolites, and we can do that by something like solid phase extraction or another method. And yet another approach that we might need to do in going from your samples to your analysis is chemical derivatization. And this is particularly relevant for GCMS analysis, where you take your metabolites and instead of analyzing them in the chemical form that they are typically in, we'll convert them to another chemical form. We'll undergo some sort of chemical reaction, and then that makes them more amenable to a metabolomics analysis on the instrument. All of that's generally handled by the core. Okay, instrumental analysis. This is the black box, or no. The goal is to use our instrument to detect and quantitate the metabolites in the sample, and there will be a lot more about that tomorrow, more than you probably want to hear. Analyzing raw data. So this is step 3 on your targeted metabolomics workflow. How are we doing on our projector? We're not here yet, so I'm on the slide that's labeled Analyze Raw Data. The goal here is to take raw data and which looks like a chromatogram. I'll show you some of that, and extract analytically useful quantitative information out of that. And the way that we do that, as I'll show you tomorrow, is by looking at the area of peaks, or the height of peaks that come out as signals on our instrument. And to a first approximation, the area of a peak in a chromatogram is proportional to the concentration of that metabolite in your sample. And we have separate signals for each of our metabolite, which is produced for us by our mass spectrometer. And that yields a table of metabolite abundances, relative abundances. They're generally, at that point, not yet absolute concentration values, and it's the next step that takes us there. And Maureen mentioned this a little bit. Oh, here we go. We're getting something at least. Great. So then the next step is to take, almost there, is to take that and do a calibration. So I'll advance so as to keep moving on. Calibration, quantitation. So here is where we go from our known quantities of metabolites to, it seems like there's some, it's a connection, because this is blinking off and on even though that we're using the new projector, so that's not a good sign. So what we're looking at here is we're looking at using quantitation by reference to a calibration curve. So we will have taken authentic standards whose concentrations are known. We will have injected them on our instrument at a variety of different concentrations. So we already know what signal corresponds to what level, just like you would do with any other sort of an assay. And we'll use the signals that we measured in the previous experiment, the peak areas from our metabolites will reference this to this calibration curve, and that'll get you your peak area. And then we'll do data analysis. And this is something that should be relatively familiar to you in terms of any other form of analysis. We'll look at graphs. We'll generate heat maps that display alterations in our known metabolites. We'll do basic statistics, t-tests, ANOVA, et cetera, things that you're very comfortable with. Most likely from your other biological work, to assess differences between samples. And there will be more on that in coming days. So that's relatively straightforward. I wanted to spend a little bit of time on the untargeted metabolomics workflow because that's a little bit more unique. Maureen will hopefully touch on that, and others will touch on that a little bit more in depth tomorrow. But the first few steps are the same. Sample preparation and instrumental analysis are the same. The next step is the major difference. Instead of looking for what we already know is in the sample, we're going to look for everything we can possibly find in the sample. We're going to look for anything that looks like it could possibly be a metabolite. Then we're going to detect those features. We'll try to align those features, find out what's in common between the samples, go back and do that again, and then we'll do our data reduction to figure out whether there's differences from group to group. And after all of that is done, we'll go in and identify our features and then do further analysis, statistics, pathway mapping, et cetera. Okay, so sample preparation and instrumental analysis. The details may differ a little bit, but in general, they're similar to targeted metabolomics, so we won't spend too much more time on that. Feature detection. So feature detection is where you attempt to find all the signals in the raw data set that originate from a metabolite. And so this is just an example of an extracted ion chromatogram. I'm sorry, a total ion current chromatogram, which just shows everything that's present in the sample. Just because it doesn't show up as a peak in that doesn't mean it's not there. Some of the features that we look at are much, much smaller relative to the peaks that are there. But we use a computational algorithm that goes through and finds anything in that data that looks like it is coming from a metabolite and puts it into a table. And then this is performed on each chromatogram, each of your samples, one by the computer. And the next step that happens is the computer aligns those features. So it says, what are the features that are in common between samples and group them together? How does it do that? Well, it looks at the information that we have from the chemical analysis. It looks at the mass. So we're doing mass spectrometry, as I'll talk about in more depth tomorrow. Every metabolite that we measure has an associated mass or mass to charge ratio, and it also has a retention time, and I'll tell you more about that tomorrow as well. That's the chromatographic information, tells you when it comes off the column. And within some degree of error, we can say that features with the same mass and the same retention time present in all of the samples are most likely the same metabolite and should be grouped together and analyzed as the same feature. So that's relatively straightforward, but when you're doing this unbiased workflow, it's an important part of the technique. So then next, we do data analysis. And so this is where we do the multivariate statistics typically. It doesn't have to be the only approach. But this is where we want to say, using all of those features that we've had, without biasing to say which one is more important than another, can we detect differences between our groups, whether we do an unsupervised or supervised, as others will describe tomorrow, and what are the components that are responsible for differentiating one group from another. And the end result of that will be a list of features which may be important in distinguishing your experimental groups. Okay, so now we have a useful piece of information that you can deal with, and you can say, you can start to interpret biological meaning into that. Okay, then and only then do we really spend much time with feature identification. And so that's not entirely true. In an unbiased metabolomics experiment performed by our core, some of the metabolites that you get out of that raw output will be identified by default. You'll already know what their names are. But some of them will not yet be identified. And more work is required either on our part or on both yours and ours to identify what they are. And so we're going to try to go from that feature using, again, the information that we already have, the mass to charge, the retention time, and any other information that we know about your sample, and additionally, sometimes other work. And we use other available resources, such as an in-house library or an online metabolite database search. Maureen already mentioned Metlin and HMDB, to try to come up with guesses as to what that metabolite could be. And these are very educated guesses. We can usually refine that list down fairly well. Sometimes there's many possibilities. And so this results in a list, either a confirmed ID in some cases, or a putative ID, a list of candidate metabolites that differentiate your group, one group from another in your sample set. And so what are you going to do with that? Well, we'll talk more about that in a little bit. The next major step is network mapping and visualization. If you decide to use that in your workflow, and we'll talk a lot more about that in upcoming sessions. This tries to put the data that you've obtained in the context of metabolic networks. Metabolites are grouped together in pathways, and flow. Metabolites flow through these pathways and are interconnected. And so visualizing alterations as they are associated with these pathways, and also looking for changes in profiles in a heat map or other means of visualizing your data can be very useful in sort of getting a handle on this very complex data. Okay, so those are the workflows that we're going to use for Untargeted. There's going to be a lot more information on all of that in upcoming sessions. Okay, so how do I decide? Maybe you already have a good idea of what you want to do. Maybe from what you've heard today, this is not much of a question. But if you're faced with making this choice, and you have a limited amount of resources, where do you choose to begin? Well, for a new project, with a sort of perhaps a general hypothesis or just a phenomenon that you want to study, oftentimes an untargeted workflow is where you have to begin, or maybe best, and it gives you a global view, whereas targeted will take you down to a more localized view later on. But there are some specific applications which favor one method over another. The biomarker discovery would certainly push you towards untargeted under most circumstances, although there are certainly exceptions. Hypothesis generation, you need to understand what may be causing a phenotype that you're observing between two animal models, or between a treatment condition and control. That may favor an untargeted analysis. If you just want to get the broadest coverage of the metabolome possible, untargeted is generally favored. Biomarker validation, targeted. So if you've gotten a biomarker from some of your studies, you may want to go to a targeted approach. If you want to quantitate metabolites known to be of interest, you've already got a defined pathway, then targeted may be what you want to do. If you need high certainty of identification of all of your metabolites, you don't want to be looking at any known unknowns, which we'll talk about in a few minutes. Targeted is best. Absolute quantitation, you're pretty much forced to be in a targeted workflow. There can be more overlap than I'm indicating here. Sometimes we can use a data set that was designed for untargeted metabolomics and mine it in a targeted fashion. But generally, you begin with one approach or the other in mind. Okay, so metabolomics versus fluxomics. There's going to be a lot more on fluxomics tomorrow. But here we have just sort of a general set of thoughts as far as, well, you've heard this technique of fluxomics. Why would I want to do that instead of classical metabolomics, and how do they differ? So conventional metabolomics measures only the concentration or the relative abundance of metabolites, the levels in a sample, what you might measure if you were doing a biochemical assay, except we give you a lot more at once. But the flux, which is the rate of turnover of a given metabolite or of a particular pathway of metabolites, is a very important aspect of cellular metabolism. And I'd submit to you that knowing the concentration of metabolites does not tell you anything at all necessarily about the flux in a metabolic pathway. It'd be seldom that you would see an alteration in flux with absolutely no alteration in metabolite levels, but knowing alterations of levels do not always necessarily give you flux. And so we can't measure it simply by observing how much of a particular metabolite is present. And so here's a picture, which would be great if you could see, that I like to use to describe metabolic flux. It's a waterfall. And so flux can be visualized as the flow of liquid between these pools in this waterfall. And the metabolite pools here, the size of these pools, are analogous to the levels of the metabolites that we would measure in a static metabolomics type of experiment. In the case of the waterfall, the direction of flow between connected pools is determined by gravity. In the case of metabolites and metabolism, it's determined by kinetics, determined by thermodynamics of reactions, determined by enzymes, many other factors. In this case, the flow between pools is not as simple as just a river flowing downstream. You can have metabolites flowing back upstream and downstream simultaneously. You can have connections in pathways that you don't predict. So it's a lot more complicated than a waterfall. But the basic parts of the analogy that help you visualize it, the amount of water flowing into the pool is equivalent to the amount flowing out of it at a steady state. But if not, then the pools have to be growing or shrinking in size. So that's a means by which flux can alter the size of the metabolite pools, what you'd measure if you did a static metabolomics experiment, but they're not necessarily directly connected. And that's what I say in this last statement here, that the size of the pool is independent of the amount of flow into or out of the pool. Okay, so tomorrow Mahmoud will tell you about how we're going to go about measuring metabolic flux. But just to extend this analogy a little bit further, what would you do if you wanted to measure the rate of flow in this river, but you couldn't put a meter in there and measure the amount of flow going down the stream? You just could take a picture and you say, I want to know how much flux that is. Well, you could come up with some ideas, but I would submit if all you had the ability to do was to measure these pools, what you might do is you might put some dye in that river. Not a good idea for the environmental catastrophe that it would be, but on an analogy level, if you put some dye in that river and you watched and you waited to see how long it took that dye to move from pool 1 to pool 2 to pool 3, you would have some idea of the rate of flow. If you already knew the size of those pools, you would have some idea of the rate of flow between those pools, and you could watch where that dye moved. Did it move to another pool down a different branch? Did it avoid a branch entirely? That would be useful information. The analog of the dye in the context of this river is an isotopic tracer. And Mahmoud will tell you a lot more about that tomorrow. A heavy isotope labeled compound that we can detect by mass spectrometry and watch it move from one pool to the other. So that's how we go about flux. I'm happy to talk more about flux, but I was requested to save some of those additional details for tomorrow to avoid some of our characteristic overlap that we have to have in a session like this. But metabolomics and fluxomics. Okay, how do I choose? Do I need fluxomics, or is it good enough to start with conventional metabolomics? For a new project, I would submit that generally jumping right in with flux, unless you have some idea of what's going to happen, is going to be a little bit too complex. start with conventional metabolomics first. Check to see if there are alterations as a first approximation. Talk with the core lab always. If there's a circumstance which pushes you towards flux right off the bat, we'll guide you in that way. But generally, that's going to give you simpler data. And as I said, although you can't tell flux by looking at levels, it would be seldom that there's an alteration in flux with absolutely no alterations in metabolite levels. So the raw data in metabolomics will clue you in that you need to do flux. Okay, so the outcome of your untargeted or your targeted metabolomics experiment will reveal whether fluxomics will be informative or whether you have enough as is. And sometimes untargeted metabolomics will lead you directly to fluxomics. Sometimes targeted will lead you. Sometimes it may be a 3-stop step to get there. But again, consult with the core lab. That is the key to doing this. So what's left in my talk from here on is examples. Since things are flashing up and on the screen, I'm still sorry about that continuing to occur. But the next part of my talk is examples of both a targeted and an untargeted metabolomics assay. There will be a great example of a flux experiment tomorrow by Michelle Wynn. So I'll briefly open it up. Any questions, anything I've left unclear about targeted, untargeted flux in general terms before I move into a few examples. I know you've heard the terms a lot today, probably getting a little bit tired of it, maybe getting a little bit epileptic from the flashing screen. But any questions I can answer before we move into that? Yes.
When you say the total level of all isotopes is just the same as level of regular targeted metabolites, if you sum all the isotopes?
Yes, I think I understand your question. And so yes, if you do fluxomics experiments, you can get a picture of the total metabolite levels by summing up all the isotopes. That is correct. However, you can't do absolute quantitation as easily because the internal standards that we use often are stable isotope labels of the same form that you may be stable isotopes of the metabolites you're trying to measure, which are often the same compounds that you may be generating in a fluxomics experiment. So absolute quantitation is trickier to do in the context of a fluxomics experiment. So there's still that advantage. Also, you're increasing the complexity of your data. So if you say, okay, I'm going to do an untargeted metabolomics experiment, and I'm going to give my cells C13 glucose before I try to do that, you're going to be making, so each metabolite may have 3 to 10 carbons in it or more, so you're going to be making, instead of a single form of each metabolite, you're going to be making 3 to 10 different forms of each metabolite. So you just increase the complexity of your metabolome 3 to 10-fold. Now there's some software which can try to ferret that out and do C13 aware untargeted metabolomics, but in general, it's making the work a lot more complicated for the computer. So especially in the context of untargeted metabolomics, we would definitely not recommend it as a first approach. Targeted, there are some occasions where you can do about as good of a job as you need to for quantitation with flux, and so you may be able to get away with a single assay, but usually you want to avoid that and start with something simpler targeted. Other questions before I move on to examples? It's a relatively long talk. Okay, let's keep going. If any questions at the end, we'll get there. So case study 1 is targeted metabolomics, and I'm going to use research that I've been working on for the past several years. I'm going to use an example in an animal model of the metabolic syndrome, and if any of you are familiar, these are our low capacity and high capacity running rats. And so just to set the research in a little bit of context, how well you run your exercise capacity determines to a large extent, or it's a very good predictor of how long you're going to live, at least at middle age and beyond. People with a high aerobic capacity as measured by metabolic equivalents tend to survive longer than those with a much lower metabolic aerobic capacity. And this is true even accounting for other risk factors for disease, like diabetes, like heart disease, etc. It turns out to be a very useful piece of information. And despite decades of study, the determinants of aerobic exercise capacity are not fully understood. And these determinants are both genetic and metabolic. And so several years ago, we set about to use metabolomics to help probe deeper into the phenotype and to the determinants of exercise capacity. And to do that, Dr. Barant and our collaborators, Steve Britton and Lauren Koch, developed. They developed the rats. Chuck initiated the research to use to develop an animal model of high and low exercise capacity in order to look at the alterations in many parts of the phenotype and that determine exercise capacity. So they took a population of genetically heterogeneous rats from a founder population and bred them. They took the rats that they ran them on a treadmill and found the ones that ran the best and the ones that ran the worst, and bred some of the rats that ran the best with other rats that ran the best, and some of the rats that ran the worst with other rats that ran the worst. Did that generation after generation, and after 10 plus generations, they had what they could call two separate lines of rats: low capacity running rats and high capacity running rats. And why are these rats interesting? They differ in their disease risk factors and other measures of bad and good health. The high capacity running rats weigh less than the low capacity running rats. They have a higher level of, the low capacity running rats have higher fasting glucose. They have lower insulin sensitivity, higher blood pressure. The HCR rats have higher VO2 max, not surprisingly, since they've been bred for exercise capacity. And they are more insulin sensitive, and they live longer. So as Chuck always likes to say, these rats eat as much or more as the HCR rats. They don't necessarily do anything more in their cages, only slight differences in basal activity, and they're healthier and they live longer. So we want to be like the HCR rats, not like the LCR rats. And how do we do that? That's what we hope to use Metabolomics to find out. Just to look at their phenotype a little bit more, they run a lot better. So this was after 30 generations of breeding or so, where they have an 8-fold difference in running capacity. This is true both in the males and the females of the species, and or of the strains. So let's look at some of the metabolic determinants of this exercise capacity difference. So put these rats on a instrument that can do indirect calorimetry and measure their respiratory quotient during exercise. And what turns out to happen is that the high capacity running rats during exercise use a lot more fat, and the low capacity running rats use a lot more carbohydrate. And the lines are longer for the high capacity running rats because as this stuff flashes up on the screen for an instant, because they last a lot longer. They have a much higher exercise capacity, and they can last longer during the protocol. So we wanted to look at that on a metabolomics context, and we began with a targeted metabolomics sort of workflow. So what we did was we took the animals and we ran them on a treadmill, or we let them rest, and we collected their tissues using the strategy that I described in my previous talk. And we did that at rest after 10 minutes of exercise, and then again at recovery. And then we did the same thing we did that with the low capacity running rats. This is close to the exhaustion time of the low capacity running rats. Then we did the same thing with the high capacity runners. Same time points, but we added 45 minutes, which is close to their exhaustion time point on our standard running protocol. And then we collected their tissues, the liver, skeletal muscle, their blood, and we ran targeted metabolomics. And so this is using the same protocol as I already described in my previous talk. And so what do we find? Well, we find there are alterations in critical metabolites. So we measured blood glucose, both using metabolomics and other conventional assays, and we find that at 10 minutes, both LCR and HCR have elevated blood glucose, but there really aren't significant differences between LCR and HCR in terms of their glucose profile during exercise, except that the HCRs drop back down to lower levels at 45 minutes. The glycogen, on the other hand, is depleted much more rapidly in the LCR rats compared to the HCR rats, and this is consistent with their increased utilization of carbohydrates during exercise. Look at the lactate. It accumulates a lot earlier in the LCR rats than the HCR rats during exercise, and this is true both in the skeletal muscle and in the blood. It's even more pronounced in the blood. Okay, so these are things you could measure classically other ways, but in the same experiment we get, and I'm just showing you a very small snatchet of the data that we've got out of this experiment, we see alterations in the TCA cycle intermediates. The HCR have higher levels of several different TCA cycle intermediates, which may be representative of an increased ability of their TCA cycle to support oxidative metabolism as compared to the LCRs. And so that was one significant difference. Another interesting difference that we observe is in the acyl-carnitine profiles. And so acyl-carnitines were mentioned once earlier today. They are a metabolite that is involved in transfer of fatty acids into the mitochondria for metabolism, and also in the transport of entities of metabolism of glucose, short-chain fatty acids. Branched-chain amino acids, and even long-chain fatty acids out of the mitochondria. And they do that because acyl-CoAs, which are the substrate that are oxidized in beta oxidation, cannot cross the mitochondrial membrane. They have to be converted to acyl-carnitines to make that transport by a carnitine transporter or carnitine palmitoyl transporter. And what we observe is that we see elevated levels of accumulation of short-chain acyl-carnitines in the LCR earlier in exercise than the HCR, which don't get there until later in exercise. And we see elevated accumulation of the long-chain, sorry, I'm pointing at a blank screen here, the long-chain acyl-carnitines in the HCR later in exercise. So this is just evidence of the increased use of fatty acid as fuel in the HCR rats as compared to the LCR. The short-chain fatty acids are indicative of something else. So we believe that the HCR rats are well suited physiologically to enhanced efficiency of oxidation of these fatty acids, and also efficiency of oxidation of the branched chain amino acids, because what we see, these short chain acyl-carnitines are actually downstream products of branched chain amino acids, and we see a buildup in these short-chain amino acid metabolism products in the LCR, suggesting that they're not clearing them effectively and not metabolizing them as effectively as during exercise. And branched-chain amino acids, looking a little further, you see that as well. The HCR rats have lower levels of exercise, lower levels of the branched-chain amino acids during exercise at 10 minutes as compared to the LCR rats. Suggesting that these are being utilized, and the LCR have a buildup of the downstream intermediates, both the short chain acyl-carnitines that I also showed you, and the long chain and these alpha keto acids, which are the immediate next step in their breakdown, suggesting that there is less efficient metabolism of these branched chain amino acids during exercise. So branched chain amino acids are interesting in a variety of different contexts. They're generally not regarded as a substantial portion of the fuel during exercise, but anywhere between 5 and 10% of total energy expenditure during exercise can be attributed to branched chain amino acids. So this is an interesting part of the phenotype of these animals. And they're also associated with other disease. There's been an association between branched chain amino acid levels and insulin resistance and obesity in humans. So the ability to more effectively oxidize branched chain amino acids may be a part, not a fully complete story yet, but may be a part of the factors that underlie, or it may either be a part of the mechanism, or it may be a sign of the improved metabolic health of the HCR animals as compared to the LCRs. So that was an interesting example in terms of the research. There's a lot more data. I can show you our paper and talk with you about it in a lot more detail. Chuck or Katie can tell you a lot more of the other pieces of information. But what's the meaning about this in terms of targeted metabolomics experiments? What do I want you to take away from that example? The majority of metabolites can be analyzed in a targeted manner using LCMS or GCMS, but not all classes of metabolites can be assayed simultaneously. We had to do at least 2, maybe 3 different targeted assays to get the data that I showed. We got more than the data that I just showed, but we had the assay that did the glycolysis, TCA, nucleotide types of stuff. We had an acyl-carnitine assay, and then we had an alpha keto acid assay reflected in that data set. Well, what if we had done an unbiased metabolomics workflow instead? We might have found that other metabolites, non-predicted metabolites, would differentiate LCR and HCR. And in fact, we do have some of that data, and I could show it at a later time, would differentiate the rats. And so that would be added benefit that we wouldn't have gotten had we started with a targeted workflow as we did. But we would have probably missed some of the metabolites in the pathways, or observed less complete coverage of some of the pathways that we were interested in, say the branched chain amino acid metabolism and all those downstream products. than we do when we go after those metabolites specifically. And the quantitation might not have been as accurate. Some of our changes were 20, 30%. And were statistically significant and biologically meaningful. If you'd done an untargeted assay, you might have gotten those changes, but perhaps not in all cases. So there's trade-offs, and that's a decision that you have to make. Case study 2, I believe someone else is going to talk about this a little bit later, so I won't belabor the biology as I may have done with the rats. But I'm going to talk about a study that we did, that I did in collaboration with Kathleen Stringer and Alex Karnowski, both of whom will talk to you later on in this workflow, about some studies in patients with acute respiratory distress syndrome, which is a condition that's observed in very sick patients, tends to be associated with sepsis and bacterial infection of the lungs. And we wanted to take a fluid, which is sometimes generated in the process of treating these patients, and look to see, in a way that's never really been done before, whether there's any meaningful metabolomic information that can be obtained. And that fluid is bronchoalveolar lavage fluid, as I stumble over the words. This is collected as effectively a means of rinsing the lungs of these very sick patients. A saline fluid is infused into the lungs of the patient and then withdrawn as sort of a wash procedure to flush out the infectious material that's been generated inside the lungs. There may be other clinical indications for this. I'm definitely not a medical doctor, but it is sometimes done in treatment of these patients. Effectively, what's generated is a wash in a saline fluid that comes into intimate contact with the lung surfaces that are the site of this infection, and what you get out is a new biofluid, if you will, that we can examine and look to see what's inside and see if what we can detect on a metabolic level tells us anything about the disease that's occurring. Briefly, some patient demographics in this study. We had disease patients and then a few healthy controls. The numbers weren't quite matched, but the differences, as we'll show, were large enough that we could see clear differences, and you can look at some other of the demographics here. Our untargeted workflow is very similar to what we already described. We extracted our samples, did peak detection, feature alignment, recursive feature detection. We had about 1,400 features from this data set. We took the top 200 differentiating features from one group to the other, and we identified and validated features and did some statistical analysis. And this is what the data looks like when you get it out of the unbiased metabolomics platform, except it's much, much larger than this. 1,400 plus features. So what are you going to do when you get something that looks like that? Well, that's the power of the computer and the algorithm that we use, and we'll even teach you a little bit about how to use on your own. This is a sort of a zoom view of the raw untargeted data. All of the metabolites have a mass to charge, they have a retention time, and they have a peak area for all of the different samples that were analyzed. There are some holes in the data. You see some missing data here, points that are not filled in with a number because that metabolite was, for whatever reason, not detected in that sample. We need a computational method that's comfortable with dealing with those things. And then we also have these known unknowns. This is sort of a silly term, but it's what we use to describe features that are consistently present in all of your samples. We know they're real, but we don't have an unambiguously assigned identity for those compounds. So the question then you would have is, are they real, or are they just some sort of artifactual garbage in my data? And what am I going to do with them? Well, the answer to that question is some are real, some are metabolites that we just don't have a name for, and some of them are effectively artifacts. Not to say that they're completely garbage or chemical noise. Instead, they may be adducts or fragments of other metabolites that we do have identified, or other metabolites that are present in your data set, whether identified or not. In other words, they're real metabolite features, but they're not unique. And then some of them are actually unique chemical species which contain biological information that we'd like to analyze. So the question is, are they worth pursuing? Do we want, obviously it would be great if we had a list with all 1,400 of these things where we could tell you exactly what all of them are, but we don't always have that luxury. The question, the answer to that question is whether they're worth pursuing is sometimes they're worth pursuing. I have a handy dandy flow chart which sort of describes our thought process on how you might go about figuring out what to do with these guys. And don't fret too much about this. A lot of this is the job of the core lab, so we will help you through this process. But it helps to know our thought process on what to do with all of these known unknowns. So the first thing to say is, are our known unknowns different between groups? If you have different groups that you're looking at, can you tell, are they statistically differentiating part of the things that statistically differentiate groups? And if they're not, my first recommendation would be to ignore it. If they are differentiating, then you may want to do some more work. A next step may be to have the core lab look at the raw data. Is this feature an adduct, or is it a fragment of another identified metabolite? If it is, if the core lab can tell you that right off the bat, then you probably want to ignore it. Alla Karnofsky has developed techniques which help you answer this question, and I'm really excited about the software that she's working on to help make this a more automatic process than painstakingly going through the data one by one and saying whether the metabolite is definitively identified and as a unique feature, or whether it's just another adduct or fragment of a metabolite. If it's not, then you can search it against an online database and say, does it have a database hit? Well, if yes, then you're in business. You have a putative hit or a list of hits that may be the metabolites you're looking at, and my recommendation would be we need to get a standard and identify it and confirm it so that you can definitively say, yes, this is a metabolite of interest, and that here's the biological meaning in the data. If not, that means you have to think about it some more. If it's really completely unknown, but it's very promising, it's the key feature in your data, then you may want to have the core lab attempt a de novo ID. The amount of times that you actually get to this point on the flowchart is very small. Most of the time you're dealing with something that's not new to science, it's just new to the method that you're looking at or new to the sample type you're looking at, and you can handle it by either one of these workflows, or by one of these three options up here. If it's not really important and promising, you may want to set it aside for now and look at the other lists on your feature as a first priority and pick it up only if you have some biological sense that it's really going to be critical. If you get to this point, you can make a paper on it almost in and of itself. But as I said, most of the time we don't get there. We can identify a metabolite by buying a standard and searching it. Okay, so an example of a known unknown in this ARDS study. So we found in our data set a large unknown peak detected in all of our samples. Wasn't in our library yet, and it had this mass to charge. What is it? Why was it in some but not all of our samples? Well, going back to something that I referenced in a previous talk, the answer we found out after some metabolite searching was it was lidocaine. This is a anesthetic that is administered to patients in some cases as part of the lavage process, and it's not administered to all of the patients because some of the patients are sedated and anesthetized using propofol or another anesthetic and didn't need this, whereas others the physician decided to use this as part of the treatment. It wasn't specified, so this was a study of convenience in some ways. The samples were already collected for another study. They were not viewing this as something that would be a likely interferent, but it shows up in some but not all of our data sets, and we found it after doing a database search, so in that case the answer was obvious. It was not of biological interest, but it was good to know it was there. In other cases, that'll come up as something of biological interest. Okay, and then we used partial least squares discriminant analysis to differentiate groups, and it shows that the groups do differentiate the healthy from the sick patients. And then we have a list of metabolites which differentiate. So which ones are those? Well, here's our workflow. We have 2,000 features. We have 3 different modes of chromatography. We did PLSDA, as I just showed you. We have our top 200 differential features. If there was no database match, if it didn't hit something that we already either knew what it was or was in one of the online databases, we didn't work with it any further. We let it aside for then. If there was a database match found, We took putative ID featured. We validated some of them with authentic standards. We re-quantitated them. This is sometimes a useful feature in sort of a targeted thing. And we found 37 now identified differentiating metabolites. If we'd gone further, if we looked deeper than the top 200, we would have probably found several more. And this was the ultimate outcome. We found a variety of different classes of metabolites, some nucleoside intermediates, some amino acids, some lipids that differentiate, and that's the outcome of the study. So we don't need to describe this in vast detail. Suffice it to say, there were various pathways that we could detect were altered by the disease. Or by the condition, and this sort of serves as a springboard for future work to define what's going on a mechanistic level. And we can take the data and we can put it on a network. This is a very simple network showing some of these affected metabolites, particularly in the purine synthesis and degradation pathway. We identified several of the metabolites that were affected in a consistent manner, suggesting that they may be involved in either the bacterial response or the bacterial load or the host response to acute respiratory distress syndrome. Key points out of this: untargeted metabolomics, the coverage gives you coverage of a lot of compounds, both predicted and unknown. Many compounds are not identified initially. You may have to do more work to figure out what's going on with those, but it can be done, and the core lab is here to help you with that. What if we'd done the targeted metabolomics first? We probably wouldn't have identified as many metabolites as we did, and quite as wide of a range of classes. We probably wouldn't have picked up on certain pathways that were altered by this disease at all. The overall data analysis would have probably been simpler and faster. We would have gotten there quicker. And we would have avoided the requantification and identity confirmation. But in this case, the ultimate outcome of the work to identify some novel pathways affected by this condition made all that extra work worthwhile. So as I've said before, it's a balancing act between those considerations. So in final summary, going way back to the beginning, metabolomics is a study of the complement of small molecule components in a system. Targeted accurately quantitates selected metabolites and classes of metabolites, whereas untargeted gives you comprehensive coverage, but at the cost of not necessarily complete identification. Again, I want to emphasize that we will give you identities with untargeted metabolomics experiment to the extent that our library is able to do so, but it will be a portion, not a complete portion, of the features that we identify. Quantitation is relative. Fluxomics uses stable isotopes, more on that tomorrow, to study turnover and selection. I'll again emphasize and double underline between the methods is best done in consultation with the core. Great, thanks for your attention, and happy to answer questions if you've got them.
In the process of untargeted analysis, your computer program seemed to go ahead and filter out things that were kind of found in both, the two different conditions. Would it filter out, say you had like a bacteriocin, it's kind of common in all your normal samples, and then a glycosylated version of the bacteriocin in your, or acylated, phosphorylated, whatever. There's a bacteria always modifying things. Mm-hmm. Would that remain or get filtered out?
So So if it's a feature that differentiates groups, however you've defined that, it will show up ideally as a differentiating feature in your list of features. But now, so you're saying, let me make sure I'm understanding. So it's something which is a metabolite that may be present in multiple samples, but it may be chemically modified in a different manner in different samples. Is that right?
Correct. That happens a lot.
So in that case, your different chemical modifications are very likely to give a different mass, right? And so to a mass spectrometer, it cares not whether it came from the same starting metabolite or a completely different pathway. If it has a different metabolite, if it has a different mass, it's going to look like a different compound to the method, and so it will be distinguished from it And if it's different between your groups, it'll pop up as something that's interesting. Whether or not something is differentiating between groups, it'll still be in that large set of data table, So if you want to say, if you were really interested in a particular class of metabolite, say your metabolite that's relevant to the infection with a bacteria or something, and you want to know what's going on and you want to make a graph out of that, you can go and look in your data set and find that metabolite, hopefully, and produce the plots and even use that to say, well, it wasn't statistically different based on this assay. So the data is there, even if it's not differentiating. In general, the workflow is such that you want the computer to highlight for you the things that are differentiating, so you can go after those as biologically meaningful targets. But yeah, no, it'll find that as long as it's within the detection limit of the instrument, and it'll be a part of your data for you to look at on your own if you want to.
What stage of the data, like you had all these different stages, what stage is it that you guys are handing back to us?
Right, so that's a great question. We hand the data back to you at, let's see if I can go back, we hand you the raw data table, and we also hand you some rudimentary statistical analysis done to, so this is not the perfect workflow. So we'll have done all of this. So we'll have done the peak detection, feature alignment, recursive feature, and we'll probably have done some basic degree of differentiation. If you tell us what your groups are, we'll put it onto a basic PCA or PLSDA analysis for you, and you can get some visualization of separation between groups. And we'll have identified those features that are present in our library. So you'll have a large data table and you'll have some basic statistical analysis. As far as identification of known unknowns, that is generally more work that we do in consultation with you. We can sometimes help you with it without doing any further analysis. Sometimes it requires more analyses. But you'll get the data back right around this stage right here. And there will be more on that tomorrow. Yes.
At this stage number 6, do you combine the data from all different platforms together before doing the statistics, or how do you combine the data from, let's say, acyl carnitines or
Sure. So it can be done whatever way that you prefer to do it. In general, we let each analysis be done separately. And it depends on the platform. If you're doing targeted, we won't do statistics for you. You should be able to handle that for the most part on your own. But if you need help, we're happy to provide it on a bioinformatics sort of scale. But in general, as I understand it, each instrument is treated separately. But it's not much of a challenge. And I think we're going to talk about Metaboloanalyst and some tools that you can use. It's not very much of a challenge to put data together and treat it in a statistical manner. What can be challenging, and what others will tell you later, is taking data from multiple batches and making sure everything is normalized appropriately, right? So if you have multiple analyses on the same platform and you want to lump it all together and do statistics on that, that can be a challenge. If you have distinct metabolites covered by distinct platforms and they all have their own levels, but they're consistent within themselves, it's actually not that challenging to put them all in the same data set and analyze them. So, if the core lab is doing some statistics for you or with a bioinformatician, I don't envision that we would have much difficulty putting it all together if that's what you preferred to do. Other questions? Great. Oh,
Thanks, Charles.
thank you very much.
Thanks for putting up with our technology.
Sorry.